Now, as we speak about the process of homeostasis, we want to think about the components. What are the players? What are the structures that are enabling this process? So we've got receptors, we've got the integrating centers, we've got effectors, and then we've got the signals. So while we've been speaking about this as an example, we want to think more relating to our body and what are the exact mechanisms that help to bring about that process, bring about that negative feedback loop. So here's a very different example. This one is regarding blood pressure, and we're going to illustrate how those four players are involved. So let's start with the receptors. So the receptors are going to be picking up or detecting any changes. Okay, so let's say you were lying down, right? You're lying in the supine position, and then you very quickly stand up, right? What happens in your body is that the blood pressure falls as a result of you quickly standing up. This is a stimulus because this is going to set off a trigger or an error signal. So the drop in your blood pressure is picked up or detected by the receptors. And these receptors are called sensors. Their specific job is to detect or sense any changes in any of the regulated variables. What they will do is send a signal, right, on sensory fibers. They're going to send a signal to the brain, specifically the medulla oblongata, and that is where we have the integrating centers. And we talked about the integrating centers last time as being somewhat of the CEO. They're going to be making all the decisions. They're usually the higher centers of the brain, and they're going to be taking in information from the sensory fibers that are bringing in those signals, deciding what type of change would be best to correct this stimulus, to correct this drop in blood pressure, and then sending those instructions to the relevant organs or organ systems that would, in fact, bring about those changes. So in this example, the medulla oblongata will pick up this change. It will send instructions to the heart on motor fibers, right? We're going to receive information on sensory fibers, and we're going to give instructions on motor fibers. And they will send instructions to the heart muscle, which will increase the rate of contraction. It's going to increase the heart rate. What is that going to do? That's going to increase our blood pressure. And so in doing that, we're going to increase blood pressure, turn off the error signal. We can consider the heart as being an effector in this loop because it's going to bring about the corrective change. And the entire loop itself would be considered a negative feedback response. All right. So blood pressure drops. We detect that change on sensors via receptors. We send those instructions to integrating centers in the brain. The integrating centers send the corrective instructions on motor fibers to the heart, and then the heart will increase blood pressure, which will turn off the original stimulus or signal. Okay. So again, some of the examples of our receptors or sensors are thermoreceptors. Thermoreceptors can detect any changes in temperature. Chemoreceptors detect changes in chemicals, right? Any chemical imbalances or chemical changes. Any neurotransmitters or ions that are present or absent can be detected by chemoreceptors. And then baroreceptors will detect changes in pressure. So any mechanical changes, right? This is one of the types of receptors that will detect changes in blood pressure. If blood pressure drops, there's less of a physical pressure on these receptors, and those will be detected and sent to the brain. If blood pressure is too high, baroreceptors would also detect that increase in pressure and would send those signals to the brain as well. So different types of receptors or sensors will detect different types of stimuli or different changes to our variables. The integrating centers here, these are going to be mostly in the brain, right? Most commonly found in the brain. And usually the medulla oblongata is the main integrating center. They're basically going to orchestrate the response. And again, I use the word CEO here because they're taking the information, making somewhat of the executive decision, and then sending the instructions down to the effector, okay? The signals are the communication. These are the messages that are being sent to and from the brain or the effector organs or even the receptors and sensors. So there can be input signals which go from receptors to the integrating centers. So from the receptors out in the periphery up to the brain, right? Those are going to be called afferent signals because they're going toward the brain. And then there are output signals which are going in the opposite direction. So those are going from the integrating center, again in the brain, down to the effector organ. And the effector organ can be the heart, but it can be a lot of other structures as well. It can be muscle, smooth muscle. It can be the gastrointestinal tract. It can be blood vessels. It can be the lungs to increase or decrease your respiratory rate. So the output signal will be going from the integrating center to the effector organ, depending on exactly where it is. Now, the signals are basically chemical messages. We'll speak a lot about this semester as far as signal transduction is concerned. How does the body actually send communication or send messages or signals? And that is basically via chemicals, neurotransmitters, or sodium and potassium movement. Those chemical movements are the mechanism by which the body will send these signals. Lastly, the effectors. So these will be responsible for bringing about the change. The various examples are glands, smooth, cardiac, and striated or skeletal muscle, as well as all of the other glands in the body, right? Your salivary glands, all of your gastrointestinal glands, those can all be areas where an effect is brought about based upon instructions from the integrating center. All right, here is a different illustration. So another way of just reiterating what we've said so far. So here's our stimulus at point 1. Here is our sensor detecting that change. It's going to send that instruction by chemical messages as input signals to the integrating centers that are in the brain. Remember, this is usually the medulla oblongata. And then the brain is going to send corrective instructions down to the relevant effector organs, and they are going to bring about the response. Now, the response ideally is going to shut off the error signal, right? It's going to go back and turn off that initial stimulus. Hence, we can consider this entire loop a negative feedback loop or a negative feedback response. And this negative attribute here, the turning off of the initial stimulus, is what makes this a negative feedback. We're going to talk coming up about some positive feedback examples, which are quite different than the actual turning off component of a negative feedback loop. At the bottom here, we've got, again, our normal range. Okay, here is our sensor being activated because we've shifted away from the set point. Here is our integrating centers sending those corrective instructions. And then here is the effector organ bringing about the change or the response, and then consequently turning off that stimulus and bringing us back to the set point. Now, a negative feedback loop, let's talk a bit more about what that is and what's entailed. So it must move in the opposite direction from the change. And this is really important because you want to distinguish negative feedback, again, from positive feedback, which we'll talk about coming up. So in order for it to be a negative feedback, it must move in the opposite direction as the change. So if the change was that the speed decreased, a negative feedback is to bring about a change that results in the speed increasing. It's going to deviate from the set point, so it's going to correct the change that deviated from the set point initially. In other words, it's going to reverse that change and bring us back to the set point value. And again here, just reiterating this idea, this is a continuous ongoing process. We're always making fine-tuned adjustments in order to maintain homeostasis. So negative feedback loops are crucial to homeostasis because they not only monitor the regulated variables to make sure that they aren't changing, or if they are changing, bring about the corrective response to reverse that change away from set point. Just driving this message home, we're going to look at another example. So another regulated variable in our body is body temperature. This one I'm sure hits much closer to home because this is one that we can actually more readily experience. So if we go out into colder temperatures, which I'm sure you're experiencing in Duluth at this point, you step outside into, 10, 15-degree weather, your body's going to feel cold, right? You're going to feel that cold. And what's going to happen is you're going to try to warm your body internally. Your body's going to bring about various mechanisms that will attempt to warm your internal organs, okay? Whether that be shivering. Okay, shivering is a great example of that, which is basically shaking or, trembling of your internal body organs and your muscles really to result in heat generation to warm up your body. Or if you go into an opposite type of temperature, into a very warm temperature or climate, you're going to experience heat and your body's going to bring about an opposite mechanism in an attempt to cool your internal organs down. So that would be more along the lines of sweating, all right? And so whether it be extreme cold resulting in shivering or extreme heat resulting in sweating, your body is trying to get you back to that regulated or get that regulated variable back to set point, which is about 37 degrees Celsius. And so that is going to generate a negative feedback loop in order to do so. Okay. Now, one of the other features of homeostasis is that there's an antagonistic effect. So this is always going to be true. And so this brings about this phenomenon called dynamic constancy, right? Dynamic constancy. So if something goes low, the body tries to bring it up high. If something goes high, the body tries to bring it down low. So there's always movement in the opposite direction from the deviation away from set point. So we can think about any number of our variables: blood glucose, blood pH, even your body weight, right? If you gain weight, your body's going to try to bring about some corrective measures, right? Like increasing your metabolism and try to decrease that back to a normal set point. If you lose weight, your body's going to try to make it harder to do so, right? It's going to bring about opposite changes that will prevent you from going lower and try to bring you back to that set point. So this is true for all of our regulated variables. And because we're moving in opposites, we refer to this as dynamic constancy. Okay, let's talk about positive feedback. And positive feedback is an opposite type of response. It is brought about to potentiate a process, to keep a process going, to amplify a certain change. And sometimes that is necessary in the body, okay? One of the main examples of positive feedback is blood clotting. So let's say you cut your finger, you got a paper cut your finger, or you injure a blood vessel somehow and you're bleeding. Initially, that blood is going to result in clotting factors that are laid down at the site of injury. And guess what? You're going to need more of those clotting factors in order to fix that injury. So what happens is you amplify that process of depositing clotting factors in order to seal that injury. That is an example of positive feedback, okay? Now, when the clot is fixed, when the damage is fixed and the clot is laid down and it's sealing that site of injury, then this will be referred back into a negative feedback and you're going to end up having the injury concealed. Okay, so positive feedback is only needed for a short time. And then ultimately, it will be ended with a negative feedback loop to close that period when that desired change is brought about. Another good example of positive feedback is the strength of uterine contractions. So during childbirth, while the fetus is moving through the birth canal, the uterine contractions need to progressively become stronger and stronger and stronger. That is an example of a desired result, which is going to potentiate that change, and will be a positive feedback. Okay, the contractions are going to get stronger and stronger and stronger until the fetus successfully is moved through the birth canal, and is delivered. Then that will be converted into a negative feedback, and the strength of those contractions will gradually decrease. So there are certain instances in the body where we do want to move away from set point for a very short temporary result or a short scenario, right? Blood clotting or a birthing process. We want to move away from set point. We want to bring about a change. And so we do that temporarily by a positive feedback, and then ultimately that will end with a negative feedback. All right. So here is the mechanism behind this positive feedback. Here's another example of this. This is one that we'll look at again when we talk about the reproductive system in AMP2. But the hormone release, which is a part of the monthly menstrual cycle in women, is a third example of positive feedback. So we have the pituitary gland in the brain secreting luteinizing hormone, that acting on the ovaries to increase estrogen secretion. And then guess what? Estrogen secretion is also going to go back and increase luteinizing hormone release from the pituitary gland. This is only going to happen for a very short period in the monthly menstrual cycle. And then it's going to be reconverted to a negative feedback whereby estrogen will decrease luteinizing hormone secretion. So I say that again to reiterate that there are times, very short periods or short windows of time where it is beneficial in our physiology to move away from set point for a desired result. And that would be considered a positive feedback. And then that would ultimately be ended with a negative feedback at the end. All right. Okay, now the purpose of having this, just to explain why this happens, is so that we can have the LH spike. So luteinizing hormone needs to be very, very high just before ovulation. And so in order to bring that about in the days leading up to ovulation, a positive feedback is initiated. And then after ovulation, this is converted to a negative feedback. And then that will continue on for the rest of the month. All right. The last thing here is talking about how we have intrinsic and extrinsic regulation, basically regulation that happens within the cells and organs to bring about change, or we can have regulation that's extrinsic where the brain gets involved. So there are some changes that are brought about initially right at the spot where we detect that change. And a good example of this is the heart. So the heart has this innate capacity to increase its force of contractility, right? How hard it's contracting, simply based upon how much volume is being filled, how much blood is returning to the heart. That would be an example of an intrinsic regulation. So if more blood volume comes back into the atria, right, the receiving chambers of the heart will contract harder. Extrinsic regulation is going to be the opposite. This is if the brain or the endocrine system or the nervous system gets involved to bring about that change. So let's look at the heart again. All right, let's say we had a scenario, which we have here on the right, where blood pressure drops and we need to increase heart rate in order to increase blood pressure. If the brain gets involved, which we know is true for this negative feedback loop, and then the instructions are sent down on nerve fibers, right, motor nerve fibers, to instruct the heart, that would be considered an example of an extrinsic regulation, because the instruction for the change is coming from a source outside of the actual effector organ. It's coming from the brain via the nerves, which would be the nervous system. So there are two different ways of bringing about regulatory changes, either intrinsically from the cells and the organs that are actually resulting in the change, or extrinsically from external factors, external instruction that's coming from the brain or the heart or the endocrine system. All right. We're almost out of time here, but I do want to go over the checkpoint questions here. So let's give a few minutes to look at these. Again, here, I'm going to invite you to write your responses down, and then we will discuss the answers. Let's give about 3 minutes, so a minute each, and then we'll look at the